Welcome back, students. So in the previous lecture, we have understood how the chain of amino acid is formed. Just for a revision, so from DNA, it will convert into mRNA. mRNA, M stands for messenger RNA. This process is called transcription, and from mRNA, it will then convert it into chain of amino acid, and this process is called translation. And in this chain of amino acid, we need to understand that the sequence of amino acid inbuilt has a code for its structure, for its native structure. And in the last lecture, we have not understood in detail how this sequence of amino acid will form. So we can do it in this lecture. Suppose, as we have seen that mRNA have 4 bases, that is A, U, C, G. Yeah, whereas in DNA, this U is replaced with T. And suppose there is a in mRNA there is a sequence like this: A, U, C, G, C, C, T, A, U, C, U, G, A. So this translation process is occurred with the help of tRNA. What tRNA does is tRNA has a specific amino acid for a specific codon. Let's just say, for example, this AUG. AUG codon will have the amino acid. AUG codon has the code. It has the code. It has the amino acid methionine. There could be a spelling mistake, but what is important is to understand the concept. This is considered as one tRNA that has this code AUG, and on the other part of this tRNA has the amino acid methionine. So when ribosome starts reading this code from this side, we will understand from which side the ribosome will read the code in future lectures. For the moment, let's assume that ribosome reads the mRNA from this side. So when it reads this, it will check whether check whether this code is related to the start codon. In order to start formation of this chain of amino acid, ribosome need a starting point, and that starting point comes with a start codon, and that start codon is defined as AUG. This is AUG. So if there is no AUG in the whole sequence, then that mRNA will never get translated. So it is essential to have a start codon in mRNA in order for this sequence to get translated into chain of amino acid and then into a protein. So we have seen that there is no start codon in this. So this mRNA will never get translated into an amino acid. So let's just change some of the sequence, and here I have written down that which sequence of bases will considered as start codon, and which sequence of bases is considered as stop codon. So now when the ribosome starts reading this sequence, when it reads this sequence, it understands that it's a start codon, and it will send a message to tRNA to bring the methionine towards it. So now in this side methionine will attach, and then it start reading the another three bases, another basis of the mRNA. We need to understand that there is a code, there is amino acid attached to every three bases of the mRNA, every three bases of the RNA. So suppose if it has a sequence like this, G, C, A, U, G. In this there is a start codon, but when the ribosome starts reading, it reads three at a time. So when it reads this, and then T should not be there in mRNA. Let's just see G. Okay. And it reads this. So even though it has the AUG, that is start codon, but it is not present at the exact point where ribosome can recognize it as a start codon. So this will not considered as a start codon. Similarly, it needs to have a stop codon, one of this thing. Like it start reading this and like this. No, it will not work out. And consider this. And now when a ribosome reads this stop codon, it understands that now it no longer need to translate further and stop the amino acid chain. So in this way, one chain of amino acid is formed. I hope at least I have been able to clear 10% of your doubts, or I have been able to provide you some knowledge. But we will continue further in the next lecture. Your comments about how to improve further the concept, and in case you have any doubts, please do let me know. Okay, have a good day.